Hello, everyone. Thank you, Neil, for the introduction and for the amazing meeting. All the talks have been so inspiring, so I hope that you also enjoy mine. I'm also here to introduce a new mouse model of AAK for investigating innovative AAK therapies. We believe that it's the most human-like AAK mouse model, and we have good reasons for that. I wanted to start with this question why we need a new model, but I think Elizabeth and Karina kind of point out to the reasons, but I would like still highlight the fact that the critical therapeutic window for aniridia, including juvenile stages that exist in human cases, is still inaccessible in mouse models. So here it is our mouse model, PAC6 haploinsufficient heterozygous mice on 129S1 SVIMJ background. I'm going to call it HET during this presentation. So this mouse model has a premature termination codon representing the most frequent human aniridia causing mutation type. And as Neil also mentioned, it was firstly reported and developed in Elizabeth Simpson's group in Vancouver, and we received the mice from them. Here we performed a deep phenotype characterization of the dynamic of AAK development in this mouse model to kind of validate its suitability to mimic the human AAK and also to unlock the inaccessible therapeutic window for investigating the AAK therapies. For this purpose, 88 mice were used and were characterized over 9 months. We used slit lamp, OCT, and IVCM. We characterized them at different 5 time points. After each characterization, 4 mice were sacrificed, 2 het mice and 2 wild type, and the corneas were preserved for histology analysis and whole man staining. So now I'm excited to share with you our findings. Here you can see a panel of the slit lamp images. The first row we have the wild type mice. You can see the transparent cornea with the full iris, which were unchanged over the time, up to 4 or 5 months. And then in the second and third row we have the het mice. Generally, we had 2 AAK phenotype, early AAK, that in the early AAK we can see the signs of the invasion of vessels very early at the age of 2 months and gradually progress to the center of the cornea over time. And in the late AAK, vessels just showed up very late, at the latest 4 or 5 months of age. And this bar chart shows that the central corneal opacity increased by age, and of course when the AAK develop over time. So yeah, in this panel you can see different abnormalities and keratopathy in this head in this mouse model. We have the keratolenticular adhesion, which is the adhesion of the cornea to the lens. It is because of the incomplete development in the prenatal period. And we had some vacuole structure. We saw it usually around the adhesion. And we had also inflammatory cells recorded in the IVCM images. Almost all of them, het mice, have these neuromas. This is the disturbed ending of the neurons. You can see here clearly the disturbed ending of the nerves. And here, these are the representative OCT images of the adhesion and the corresponding HN staining and how do they look like. In the histological analysis, we can see that epithelium in het mice were significantly thinner than the wild type. The columnar structure of the basal cells is gone in het mice, and resulting in flattened basal cells. As you may know, basal cells build a basement membrane by producing components like collagen 4. For this reason, we investigated through the double staining for collagen 4. And interestingly, in the het mice, we can see that a loss of collagen 4 expression in the basement membrane. After applying fluorescent dye in the live cornea and imaging under blue light, it further confirms the impairment of the barrier function in the epithelium. We did further analysis in het mice in terms of the epithelium thickness, epithelial layers, and epithelial layers thickness, and in all cases, in het mice were significantly lower than the wild type. And here also we have the representative images, OCT images, that we show that anterior chamber depth is significantly lower in het mice compared to the wild type, which is aligned with the human studies as well. Progression and severity of AAK was assessed with the slit lamp images, slit lamp by microscopy, to visualize the limbal region in all mice, and clinical grading scale for AAK was applied to all of them. Here in the first row, we have the wild type, and the rest are the het at different grades. As you can see in the wild type, we have the transparent cornea with the completely intact limbal border, and the Holman staining for CD31 also confirmed that the vessels were confined to the peripheral, to the limbal border. All the mice at the age of 1 month had grade 1 or 2, with the vessels just bleaching the limbal border, invading the peripheral cornea. You can follow up the trend in the Holman staining. As mice age and AAK progress, vessels invade totally into the center of the cornea, here, and in the Holman you can see the vessels. To investigate the impact of AAK development on the corneal epithelium, we used IVCM to image each layer of the epithelium, including superficial epithelium, wing cell layer, basal cell layer, stroma, and endothelium. This panel is so informative and have so many details, and we have already discussed everything in the paper that is going to be published soon. But in total, taken together, the results indicate a progressive breakdown of the cellular and neural structure of the corneal epithelium and stroma in the het mice, and it gets worse as the AAK develop to the grade 4. To characterize corneal neovascularization with AAK development, whole mount corneal tissues were immunostained for CD31 as a marker for blood vessels, and the live one as a marker for lymph vessels. The first column, we have the wild type mice. The cornea maintained corneal angiogenic privilege devoid of lymph vessels and blood vessels. But surprisingly, at the age of 1 month in het mice, an early and selective ingrowth of corneal lymph vessels were observed. These lymphatic vessels not only did not regress, but also persist up to at least 4 or 5 months of age. CD31 Holman staining revealed that corneal hemangiogenesis was less prominent and was delayed relative to lymphangiogenesis. We also did some immunostaining on paraffin sections of an vascularized eye, and this staining also confirmed that lymph vessels were accompanied with the F480 positive macrophages. We all know that these macrophages are necessary, play an important role for in the inflammation and inducing lymphangiogenesis. We also performed beta-3-tubulin whole mount immunostaining to investigate the distribution of the nerves. The first row we have the stromal nerves, and the second row we have the subbasal nerves. The first column is the wild type, and as you can see, in the wild type, stromal nerves were confined to the peripheral. However, in the het mice, they were extended to the central of the cornea. And in the stop-basal nerve, the fiber nerves in wild type were densely distributed and organized centripetally, forming a vortex at the corneal apex. However, in het mice, we have an uneven and sporadic distribution of subbasal nerves, and in this case, you can see that a stromal trunk kind of invade the subbasal nerve layer, confirming that the loss of stromal epithelial compartmentalization of the nerves. And at the last, as you all know, the emergence of AAK coincides with the progression of the limbal stem cell deficiency. So we decided to investigate the distribution and location of the limbal stem cells in wild type mice and in het mice with grade 1 and 2, and also in het mice with grade 3 and 4 to be able to compare them. The two first row belongs to the wild type, and the rest are the het. Here we have the het mice with grade 1 and 2, and here we have the het mice with grade 3 and 4. As you can see here, we have the P63. We use P63 as a well-known marker for limbal stem cells, and GPHA2 as a novel marker for limbal stem cells. And here, as you can see in het mice, P63 expression was confined to the single layer of the basal cells in het mice compared to the wild type. But interestingly, even at the later stage of AK, we still have the P63 positive cells in the basal layer, although a further loss of P63 expression is apparent. P6 and PAC6 and keratin 12 also followed the same pattern in the grade 1 and 2 and grade 3 and 4. However, it's kind of different about GPHA2. As you can see in grade 3 and 4, it seems that GPHA2 expression was kind of shifted to the superficial epithelium. So we think that GPHA2 expression in the basal epithelium appears to mirror the progressive limbal deficiency, and it would be an important marker for further evaluation in the context of aniridia. And you may ask that what are these population in this in the stroma? This row show the attachment. These are the remnant material of the lens, resulting from the incomplete development in the perinatal period, and we were surprised that they were also positive for limbal stem cell markers. So we guess that they are kind of stem-like cells. And at the end, after sharing these findings, I hope you are also convinced that this model is the most human-like AK mouse model, and it can also kind of unlock the therapeutic window for evaluating targets interventions for later studies. I have heard many talks during last few days about the human studies and the in vitro studies, and I was kind of surprised that all the findings has a lot of overlap, and it shows that kind of convinced that we are on the correct path to find the solution and treatment. Thank you.
Very nice, Dina. Amazing amount and quality of data. Congratulations. About the nerves, this just popped in my head quickly. Do you see invasion of the nerves in the central? Yeah. Do you see more nerves? Can you quantify it somehow? What's the feeling even?
You mean to be able to count them?
Just because I see in the cells, in the iPS-derived cells, I see like this sort of neuronal signature that the aniridia cells seem to have compared to the control. And now you show more nerves. I mean, is this related somehow?
Yeah,
No, not at all. Do you have any idea? Could you tell like there's a lot more, or are they just longer?
Yeah, that's actually good point, and we kind of intended to count the nerves, so it can be a good idea to investigate more.
Okay. I was just wondering if there could be any correlation, but yeah.
Yeah, there could be.
You can think about it.
Good point, thank you.
or overexpression of those markers could somehow be the disruption of the nerves.
Hey, Dina, that was great, absolutely great. If I understood correctly, with the two markers you used, you saw stem cells, or limbal stem cells in quotes, right to the end of the latest time that you've looked at. What is your thinking about that? Are those markers biologically suggesting that those cells are just sitting there but not doing anything? Or what is your thinking about that?
Actually, I think it just highlights the fact that we should focus more on the niche preservation of the limbal stem cells. We have the stem cells there, so maybe it's because of the loss of the different factors that are involved in preservation of limbal stem cells. So if we can recreate the niche, they can be productive or able to regenerate the cornea again.
Thank you.
Or perhaps even as we've seen like yesterday, that maybe these cells still are there, and maybe they still are there even in the human cases, but maybe they are not able. They can't be activated in the same way as
True.
and maybe so it could be interesting, that to think that the stem cells are still there, but somehow we know they're insufficient, but yeah, maybe they're just not activated in the right way. Okay, if there's no more questions, any more questions? No? Okay, so if there's no more questions, thank you again, Dina.